Hello students, how are you all? I hope you all are doing fine. Welcome to One Page Biology. In today's video, I will be covering a very important topic of 12th standard NCERT as well as the boards. The name of the topic is transcription. Dear students, this particular topic is part of the chapter Molecular Basis of Inheritance, which is there in NCERT unit Genetics and Evolution. The same chapter Molecular Basis of Inheritance is also there in your 12th standard board textbook. So dear students, let us try and understand what is transcription. Hello students. So let us try and understand what is exactly transcription. Dear students, the word transcription comes from the word transcript, which means mainly information. And what exactly is this information about? This information is mainly of the proteins and where it is present. Where is this information of proteins present in the cell? Dear students, you must have guessed it right. The information is present inside the DNA. Now in the background, can you all see the red color strand on the screen? So this particular red color strand is nothing but our DNA, which is the genetic material. And this DNA we all know is present inside the nucleus. What does this DNA contain? The DNA contains the information of proteins. This information of proteins is actually passed from DNA to the RNA. So actually DNA, which we all know is the main genetic material in every organism, whether it is bacteria or plants or animals, in every organism DNA is the main genetic material. So the DNA contains the information of proteins, and this information of proteins is actually transferred from DNA to the RNA, and this process of transferring the information from DNA to RNA is called as transcription. So let us understand how does this occur. We just now said that DNA, which is the main genetic material, it contains the information of proteins. And where is DNA located? As you can see on this right hand side, it is present inside the nucleus of the cell. So DNA contains the information of proteins, but this information is passed on into the RNA. And by which process, dear students, it is rightly said as transcription. But dear students, there is one more process which is called as translation. And what is translation? So this information now, once it is passed into the RNA, that information is now translated in the form of actual protein, and that process is called as translation, wherein the information of RNA, which is of protein, is actually converted into protein form. Now dear students, where exactly is transcription occurring? Now as you can see on the screen, in case of bacteria, generally bacteria are nothing but prokaryotes. So there in them the transcription process is occurring inside the nucleoid, because as you all know that bacteria do not have a well-defined nucleus. What they have is actually a nucleoid, which is simply the DNA. So dear students, and you can see in the screen, basically this is what is the DNA of a bacteria, and this is also called as a nucleoid. Now in case of eukaryotes, the process of transcription is occurring inside the nucleus. And we all know the nucleus is well developed, well defined in case of eukaryotes. As you can see in the screen, the nucleus is present, which is a well developed nucleus. Now going ahead, where exactly, when does this transcription occur? Dear students, that's the big question, that when does this transcription occur in the cell? Now dear students, as you must be aware that there is something called as cell cycle. And what is cell cycle? Cell cycle is nothing but a set of changes which actually occur inside the cell when it basically undergoes division. So for that we need to understand cell cycle in details, and I will be coming with one of the videos on cell cycle separately. But as of now, please try to understand that the process of transcription is occurring in two basic events inside the cell cycle, and these are nothing but G1 and G2. So as you can see in this diagram, this is indicating the G1 phase and this is the G2 phase, right? So it is in this particular phase that the transcription is actually taking place. Now, how does this occur, dear students? How does the exact transcription process occur? So as we said that transcription is nothing but the process in which the information is getting transferred from DNA into the RNA. So as it has been mentioned over here that it occurs on the segment of DNA. That means from DNA RNA is going to be formed. Correct? So DNA is how many stranded? We all know it is a double stranded structure. And that segment of DNA which is actually going to form the RNA, that segment of DNA can be called as transcription unit. Now let me show you the picture of it. So for example, this is the transcription unit, dear students. So in this picture you can see that there are two strands, right? These are the two strands of DNA, wherein one strand is in the direction of 3' to 5' end and the other strand is in the direction of 5' to 3' end. As you all know that the DNA is basically anti-parallel in nature. Dear students, if you have not watched the video of DNA replication, please watch the video of DNA replication which I have already made. So coming back to the topic, as we said that from this DNA the RNA is going to be formed. But now dear students, we need to know that there are some sequences which also help in the process of transcription. There are two main sequences which also take part in this process. That is, the initial sequence is called as the promoter sequence. Now as you can see here, the promoter is mainly present initially at the start, at the beginning. So what is exactly the promoter? Now the promoter sequence is mainly the sequence which starts the process of transcription, right? It starts the process of transcription. Whereas there is a sequence which is after promoter which is called as a structural gene. What is a structural gene? Now structural gene is the part of DNA, dear students, which actually contains the information of protein, right? I said promoter starts, or it is a sequence which starts the process of transcription, whereas structural gene is the part of DNA which contains the information of protein. And then comes the last sequence which is called as terminator. From the word itself you can guess that terminator means to terminate, means to stop. So this is the sequence which will stop the process of transcription. So dear students, you have to remember there are three main units or three main units of transcription. One is nothing but a promoter which is present in the beginning. The second is structural gene and the third one is terminator, right? So this entire unit, entire segment of DNA which consists of a promoter and a structural gene and the terminator is called as, basically it is known as the transcription unit. Now what exactly is template strand, what is a coding strand, I will explain you in the coming slides. So now talking about the entire process of transcription, it is divided into three steps, dear students. One is called as the initiation, second is elongation, and third is termination. Now what is initiation? Initiation itself means the beginning, that means the start of transcription. Elongation means the formation of RNA on the DNA, right? That will be called as elongation. Whereas what is termination? Termination will be the end of transcription. Termination will be the end of transcription. So going ahead, let us understand first what is initiation. Now as it has been mentioned that in initiation the RNA polymerase binds to the promoter. Now dear students, what is exactly RNA polymerase? Now dear students, RNA polymerase is nothing but an enzyme which is required to form the RNA from DNA, right? So there has to be an enzyme which helps in the formation of RNA. Now what is this RNA polymerase going to do in the beginning of transcription? So in the first step, that is initiation, as you can see on the screen, in initiation RNA polymerase enzyme binds to the promoter, because remember we had said that promoter is present before the structural gene. So basically from here the process of transcription is going to begin, and that's why, as I said, that RNA polymerase is going to go and bind to the promoter. Now dear students, there is one important thing which we need to know over here. This particular enzyme called as RNA polymerase, it is a kind of a complex enzyme. Why is it complex? Now in case of bacteria, as you can see on the screen, I have mentioned that in case of prokaryotes means bacteria, right? So in case of prokaryote, this same enzyme is made up of 5 units. 5 units. Can you count the 5 units? That is, there are 2 alpha units, 2 beta, that is one is beta, another is beta dash, and one is omega. So these are the 5 basic units of the enzyme, which is RNA polymerase, which is going to help in transcription. Now what happens is, this enzyme, as we said, is going to bind to promoter, right? In the previous diagram we said it is going to bind to the promoter. But how does it bind to the promoter? Now dear students, this RNA enzyme, in case of bacteria, with the help of a sigma factor, there is something called as a sigma factor. What is a sigma factor? It is a protein mainly. So this sigma factor helps the RNA polymerase to bind to the promoter. Am I clear with that? Now you may wonder, how does this happen in case of eukaryotes? Now dear students, RNA polymerase of eukaryotes is slightly different. Now going ahead, dear students, once the RNA polymerase binds to the promoter, now it will cause the DNA to unwind, or you can say it will separate the 2 strands of DNA. Now as you can see on this, students, can you all see that the 2 strands have separated? Yes. So once the 2 strands separate, the nucleotides on both the strands have been exposed, right? Now the question is, on which strand the RNA is going to be formed. Now please, dear students, for that you have to understand a concept, that is, basically once the RNA polymerase unwinds the DNA, it forms the structure. Can you see a structure like this? It is looking like a bubble. Yes, dear students, so this bubble is called as a transcription bubble. So first the RNA polymerase unwinds the DNA, it forms the transcription bubble, and now there are 2 strands on the DNA, that we can say that one strand which is in the 3' to 5' direction, other strand is 5' to 3'. Now the strand which is 3' to 5' direction, dear students, is nothing but the template strand. Now on this particular strand the RNA is going to be formed. Dear students, you have to remember this very clearly, because on this there are many questions which are asked in the NEET as well as in your board examinations, that on which strand the RNA is formed. So please remember, it is the template strand. And which is the template strand? The strand which is from 3' to 5' direction. Whereas the opposite strand or the complementary strand of DNA, which is from 5' to 3' direction, it is called as the coding strand, right? So as in this particular picture, the strand which is from 5' to 3' is nothing but the coding strand, right? Okay, now on the template strand down here, you can see that on the template strand which is in the 3' to 5 direction, the RNA is getting formed, right? So now, dear students, the RNA polymerase one by one will start bringing the nucleotides, and it will start joining them as according to the complementary rule, and that's how the new RNA strand is going to get formed. So if I ask you the question, in what direction the RNA is formed, the direction is 5' to 3'. On which strand the RNA is formed? It is on the template strand which is from 3' to 5'. But remember, dear students, the RNA contains uracil instead of thymine, right? So wherever you can see on this particular RNA strand, in between there are uracils. So basically the uracil is going to pair with the adenine, because in case of RNA there is no thymine, there is uracil instead of thymine. Okay, and the complementary rule is always that adenine will now, according to RNA, it will pair with uracil, and guanine will pair with cytosine. Again, if you haven't understood the complementary base pairing rule, please check out the video of DNA replication. Okay, so now let's see. So RNA is going to get formed as per the complementary rule, and this process will keep on continuing. So this process of formation of RNA, that is in direction, that is which direction it is getting formed, 5' to 3'. So RNA polymerase keeps on joining the nucleotides, and this process will keep on taking place. This entire process of formation of RNA is called as elongation. That means the RNA is getting formed. This yellow part is mainly indicating the RNA polymerase enzyme, and as soon as this RNA polymerase enzyme reaches to a particular site on the DNA which is called as a terminator site, right? So there are terminator sites in case of bacteria, there are terminator sites in case of plants, animals. So as soon as the RNA polymerase enzyme reaches to the terminator site, what will happen? This RNA which has been formed, dear students, it is going to get released. RNA is going to get released in the nucleus of eukaryotic cell. Okay, but now in case of prokaryotes, dear students, remember there is one factor also which helps in the termination, and this particular factor is called as Rho factor. So Rho factor helps the RNA polymerase to recognize the terminator site. So for example, if this is a DNA, right? So the RNA polymerase enzyme, so the yellow part is indicating the RNA polymerase enzyme in this case. So as soon as this yellow part will reach to the terminator site, and how will it recognize the terminator site? The Rho factor will help in recognition. As soon as it recognizes the terminator site, the RNA polymerase will stop the formation of RNA, and this particular RNA, as you can see, will be released with the help of Rho factor. At times, guys, there are also certain poly-U sequences on the DNA which also help in the termination of transcription, because of which there is a formation of a hairpin loop-like structure in the RNA. But we won't get into that much details, right? So what you need to know is basically there is a terminator site which is after the structural gene, and RNA polymerase, as it reaches to the terminator site, it will stop the process of transcription and the RNA will be released in the nucleus. We all know that RNAs are of three types, which are nothing but mRNA, rRNA and tRNA, right? So mRNA, which is nothing but messenger RNA, rRNA is ribosomal RNA, and tRNA is transfer RNA. But out of this, the RNA which mainly contains the information of proteins is the mRNA, and mRNA stands for messenger RNA. Now if you compare the prokaryotic mRNA and the eukaryotic mRNA, dear students, there is one important difference. In case of prokaryotic mRNA, there are no non-coding sequences which are called as introns, whereas in case of eukaryotic mRNA, there are non-coding sequences as well as coding sequences. Now, non-coding sequences are called as introns and coding sequences are called as exons. So for example, in this particular diagram, as you can see the DNA, this is a DNA of a eukaryote, right? So in this case, the blue part is indicating the non-coding sequences which are called as introns. So they are actually of no use. So once the mRNA is getting formed, these introns are going to get removed finally, and finally what we get is a mature form of mRNA. But that process is called as processing of mRNA, and that we will be studying in some other video. So now, dear students, what we need to know is the introns are absent in prokaryotic mRNA, whereas they are present in the eukaryotic mRNA. Now, as we said that there are three types of RNAs which are going to be formed: mRNA, rRNA and tRNA. The RNA polymerase helps in transcription. So in case of prokaryotes, there is only a single RNA polymerase which forms all the three types of RNA, that is mRNA, rRNA and tRNA. Whereas in eukaryotes, there are three separate RNA polymerase enzymes which actually help in the formation of. So for example, as you can see that for the formation of rRNA, that is ribosomal RNA, there is an enzyme called as RNA polymerase 1. For the formation of mRNA and snRNA, now here what is snRNA? snRNA is small nuclear RNA. Both these RNAs are formed in case of eukaryotes by the enzyme that is nothing but RNA polymerase 2. And for the formation of 5S rRNA again and tRNA again is transfer RNA. For which enzyme is helping? It is RNA polymerase number 3. So you need to know that there are three different RNA polymerases for the formation of three different types of RNA in case of eukaryotes. So dear students, I hope you have understood the process of transcription. Thank you. So how was the topic, dear students? I hope you all have understood transcription. If you have understood the topic, please hit the like button as well as share it with your friends. And also please let me know if you have any doubts in the comment section. I will be coming with more and more videos for you all related to biology. Till that time, take care. Thank you.